Hello to everyone, my name is Almudena Neva-Alejo and I'm going to talk about sex differences in Alzheimer's disease. Alzheimer's disease is the leading cause of dementia. The number of patients affected by this disease is growing, and despite the effort made by the scientific community, we still don't have efficient treatment and precise diagnostic method. That's why it's important the investigation in this field. Since sex differences in prevalence, severity, and progression of Alzheimer's disease have been reported, the inclusion of sex perspective in biomedical research could help to a better and faster understanding of diseases. For these reasons, the main objective of this work is unveiling sex differences in Alzheimer's disease through an integrative single-nucleus transcriptomic in silico approach. The methodology can be summarized in three main steps. The first one is a systematic review to select those studies that we are going to analyze from public repositories. The second one is the individual bioinformatic analysis, and the third step is the integration of individual results and the biological interpretation. In the individual analysis, we downloaded the single-nucleus RNA-seq data from the different studies and did some steps to process and explore the data, like quality control, normalization, or dimensionality reduction. Finally, we use this contrast to do a differential expression analysis by cell type, in particular in microglia and astrocytes, to detect sex transcriptomic differences in Alzheimer's disease, and a differential abundant analysis to detect sex differences in cellular composition in Alzheimer's disease. Here we can see a diagram that summarizes the systematic review. Basically, we apply some inclusion and exclusion criteria that afforded the selection of 3 studies. Then, in this plot, we represented the percentage of mitochondrial genes per cell and per patient before quality control. This covariable is a quality control metric because a high percentage of mitochondrial gene is a sign of low quality cell or nucleus. In this case, we have used data from single nucleus experiments, so we shouldn't find mitochondrial genes in our data, and that's why we eliminate the patient number 6 from our analysis. After analyzing dataset individually, we integrate all the results. In this table, we can see the result of the integrative analysis. We detected 6 sex-specific differentially expressed genes in microglia and 59 in astrocytes. Also, we detected 1 non-sex specific differentially expressed gene in microglia and 20 in astrocytes. The 40% of detected genes had a previous described association with Alzheimer's disease, providing robustness to our analysis. About the differential abundant analysis, the result didn't show sex differences in cellular composition in Alzheimer's patients. Finally, the conclusion. First, single-cell and single-nucleus transcriptomic approaches have great potential for the study of human diseases, as they can reveal cellular heterogeneity masked in bulk RNA-seq results. Then, this study has allowed the characterization of sex-specific and non-sex-specific transcriptomic differences in microglia and astrocytes in Alzheimer's disease. This supports the need to include the sex perspective in biomedical research, being a fundamental step toward personalized medicine. Finally, some detected genes confirm other study conclusions, while others provide new research lines that could help to understand this complex disease, as well as improve its treatment and diagnosis. And that's all. Thank you for your attention.